Hi, this is Miss Vitale. This podcast is intended for AP Bio. It is a unit on cells and it corresponds with Chapter 6. In the 1600s, Robert Hooke looked at cork under a microscope, and he named the empty boxes he saw cells after the tiny rooms in monasteries, much like the cells in a prison. He produced a book in 1665 called Micrographia with over 60 images of cork magnified. He never studied living materials in which the content of the cells could be seen. Anton von Leeuwenhoek was making powerful microscopes about the same time. He was a Dutch drapery store owner who ground lens as a hobby. He was the first to observe living organisms in pond water. He didn't know he was observing microorganisms, and he drew no conclusions about the cellular nature of things. Since the days of Hooke and Leeuwenhoek, improved microscopes have vastly improved our view of life at the cellular and subcellular level. Light microscopes pass visible light through a specimen. You can see very large bacteria with a light microscope, but no detail. Their total magnification only reaches 1,000 times without causing blurriness. The eyepiece is 10x, and the objective lens ranges from 4 to 100. You multiply the 10x of the eyepiece with the power of the objective lens to get your total magnification. A micrograph is a photograph taken through a microscope. Magnification increases the apparent size of an object, and resolving power measures the clarity of the ability to show two objects as separate objects. Electron microscopes were invented in the 1950s. They use beams of electrons instead of light. They have a much higher resolution and magnification. The more powerful electron microscopes can see objects as small as 0.2 nanometers. A nanometer is 1/1,000,000th of a meter. A printed period is about 1 million times bigger than 0.2 nanometers, which is the size of a large atom. Only under special conditions can an electron microscope see an atom, but they can see organelles and molecules such as DNA. An SEM is a scanning electron microscope, and it sees 3D details of the cell surface. Electron beam scans the surface of a cell that has been coated with metal. That stops the beam and emits electrons. TEM is a transmission electron microscope, and it sees internal structures. Specimens are sliced very thin, and electron beams pass through the thin layer. One of the big disadvantages of an electron microscope is that the specimen has to be in a vacuum with no air or liquid, so you cannot observe living things. That, however, is an advantage of the light microscope, which can be used to observe living cells. Scientists can study individual organelles by centrifuging them in a high-speed centrifuge called an ultracentrifuge. They use cell fractionation, which is basically putting cells in a blender, chopping them up, spinning them in the ultracentrifuge, which separates out the larger particles from the lighter particles. The largest particles, or the heaviest particles, sink to the bottom, and you wind up getting layers of the different organelles so they can be studied individually. The smallest cells are bacteria called mycoplasms. They range from about 0.1 to 1.0 micrometers. Again, that's 1/1,000,000th of a meter, or 10 to the minus 6. The largest cells are bird eggs and some nerve and muscle cells. Most cells range between 1 micrometer and 100 micrometers. Cell size and shape are related to function. Bird eggs are bulky because they contain large amounts of nutrients. Long muscle cells pull different body parts together, and red blood cells are tiny to fit through capillaries. At a minimum, a cell must be able to contain enough DNA, protein, and internal structures to survive and reproduce. The maximum size of a cell is limited by its requirement for enough surface area to take in nutrients and dispose of waste. Large cells have more surface area, but less surface area per volume. Prokaryotes include bacteria and archaea. They have no membrane-bound nucleus. They are very small, 2 to 8 micrometers, about 1/10 the size of other cells. You need an electron microscope to see the structures inside of them. DNA is coiled into a nucleoid region like a nucleus, and it has no membrane. The plasma membrane used to be called the cell membrane, and it surrounds the contents of the prokaryotic cell, which separates the contents of the cell from the environment. It also controls what goes in and out of the cell. The prokaryotic cell wall is made of a complex called peptidoglycan, and it varies from bacteria strain to bacteria strain. Peptidoglycan is a combination of sugars and proteins. It is outside the plasma membrane, and it is rigid and chemically complex. It protects the cell and maintains the shape. Some bacteria have a capsule. It's a sticky outer coat. It adds further protection, and it also can help bacteria stick to things like surfaces and tissues. Some bacteria also have pili, which are short projections for attaching things. Flagella is found in a few strains of bacteria, and it is a long projection for locomotion. Eukaryotes include animals, plants, fungi, and protists, and they have a membrane-bound nucleus. They are more complex than prokaryotes. They are larger and have a bigger variety of structure. The cytoplasm is the fluid-filled region between the cell membrane and the nucleus, and the cytoplasm is made of a gel-like substance called cytosol. Organelles are structures with a special function within a cell. Most are membrane-bound compartments. They do different metabolic processes. Therefore, a lot of different processes can occur at once in a cell in the different compartments. There are some non-membranous structures like centrioles, flagellum, and the cytoskeleton. The nucleus is the genetic control center of the cell, and it contains the DNA. Chromatin is a stringy form of DNA and proteins. Chromosomes form during cell reproduction, where the chromatin condenses into tight coiled rods. Humans have 46 chromosomes in 23 pairs. Chromosomes are visible with a light microscope during cell division. The nuclear envelope is the membrane surrounding the nucleus. It is a double membrane that has pores that control what material goes in and out of the nucleus. In addition, a nucleus may have a nucleolus, which appears as a dark spot. There could be more than one on a single nucleus. It is a mass of fibers and granules made of chromatin, DNA, RNA, and proteins, and it is where ribosomes are made. Eukaryotes have organelles that are combined and connected to each other with an internal network of membranes. The endomembrane system is that cytoplasmic network of membranes. It divides the cell into compartments and surrounds the organelles. The membranes participate in metabolism and may contain enzymes. By dividing the internal environment of cells, many processes can occur at once. The endoplasmic reticulum is a prime example of the interrelatedness of parts of the endomembrane system. Endoplasmic reticulum is Greek for a network within the cell. The cisternae is the space created by the membranes of the endoplasmic reticulum. Smooth ER lacks ribosomes. It has enzymes embedded in the membranes that synthesize lipids like fatty acids, phospholipids, and steroids. For example, the smooth ER in the ovaries and testes of mammals make the sex hormones estrogen and testosterone. Liver also has large amounts of smooth ER, and it contains enzymes that regulate the amount of sugar released from the liver into the bloodstream. There are also other enzymes in liver cells that help break down drugs and harmful substances like barbiturates, amphetamines, and some antibiotics. When you take too many drugs, smooth ER and enzymes increase in your liver cells, therefore increases the body's tolerance, so you need more and more of the drug to achieve the original effect. Smooth ER can also store calcium ions. In muscles, the calcium ion is necessary for muscle contraction. The nerve signals the muscle cell. The calcium leaks from the ER into the cytoplasm of the muscle cell and triggers a contraction. Rough ER is studded with ribosomes. Ribosomes produce proteins. The ribosomes that are free-floating in the cytoplasm produce proteins for the cell. The ribosomes that are studded, the rough ER, make proteins that are excreted by the cell. Those are called secretory proteins. For example, antibodies are a defense molecule. They are made and secreted by white blood cells. The ribosome makes the polypeptide. Short chains of sugar link to the polypeptide, creating a glycoprotein. That is packaged into a transport vesicle and goes to the Golgi apparatus for more processing. The Golgi apparatus was named after an Italian biologist named Camillo Golgi. It is made of flattened sacs like stacks of pita bread that are not connected. The number of Golgi stacks depends on how much protein the cell secretes. It performs in partnership with the ER. The Golgi apparatus receives and modifies substances produced by the ER. One side of the Golgi stack receives transport vesicles from the ER. It takes the material and chemically modifies it. That marks and sorts it for different destinations. It then repackages it into new transport vesicles to export out of the cell or to become part of the cell.
Two membranous organelles, the Golgi apparatus and the endoplasmic reticulum, or ER, are the main components of the cell's endomembrane system. A primary function of these organelles is to produce mature proteins that will be exported from the cell, incorporated into membranes, or moved into other parts of the endomembrane system. For simplicity, we'll describe these organelles from a two-dimensional point of view. New proteins made in the ER either enter the lumen of the ER or are embedded in the membrane. Inside the ER, enzymes add sugar groups to some of the proteins, creating glycoproteins. The ER directs its proteins to the Golgi apparatus. Transport vesicles bud off from the ER, taking with them a cargo of membrane and lumen proteins. The vesicles fuse with the Golgi apparatus, adding to the Golgi volume and protein content. The sacs of the Golgi apparatus are called cisternae. The part of the Golgi apparatus that lies nearest to the ER is the cis region. The part nearest the plasma membrane is the trans region. The middle cisternae make up the medial region. In one model for protein transport within the Golgi, the proteins are delivered in vesicles from one compartment to the next. Each compartment contains Golgi enzymes that perform modifications to the proteins. For example, in this particular sac, a sugar residue is cleaved from proteins. Mature proteins in the trans region of the Golgi are packaged into vesicles in preparation for travel to their final destination. In this case, proteins are targeted to the plasma membrane for secretion outside of the cell.
Lysosomes are produced by the rough ER and the Golgi apparatus. They contain digestive enzymes in that membrane sac. Lysosomes can digest old cell parts, bacteria, and other things that are no longer needed. The rough ER produces the transport vesicle with the inactive hydrolytic enzymes in it. The enzymes enclosed in the lysosome so they don't digest the cell. They go through the Golgi apparatus for processing, and then the lysosome is produced. The lysosome hooks up with a food vacuole that is formed when a particle of food enters the cell. The food vacuole merges with the lysosome, and the enzymes inside the lysosome digest the food. For example, if this was a protein, it would be digested in the lysosome into individual amino acids, and then those amino acids could be used by the cell to build its own proteins. Lysosomes are also used in the development of animals. As an embryo is developing, the webbing that joins the fingers in early human embryos is eliminated and digested by lysosomes so the individual fingers can form. In addition, as a tadpole is metamorphosizing into a frog, the tail of the tadpole is digested by lysosomes. Lysosomal storage diseases are hereditary disorders. They result from missing a hydrolytic enzyme in lysosomes. What that causes is the lysosome to become engorged with undigested substances. There was a movie called Extraordinary Measures with Brendan Fraser and Harrison Ford about Pompeii's disease. This was when large amounts of glycogen accumulate in the liver cells because there are no glycogen digesting enzymes. Tay-Sachs disease is common in Jewish people, and they lack a lipid digesting enzyme in their lysosomes, so the nerve cells in the brain accumulate lipids and are damaged. These children die very young. Vacuoles are membrane-bound sacs. They are larger than vesicles. They come in different shapes and sizes and have a variety of functions. I already mentioned food vacuoles that work in conjunction with lysosomes. Plant cells have a large central vacuole that can serve as a large lysosome. It also may help the plant grow in size by absorbing water. It can sometimes store chemicals or waste. For example, the central vacuole in flower petals contains pigments that attract pollinators. Other central vacuoles of plants can contain poisons that protect them against plant-eating animals. Some protists have different kinds of vacuoles. For example, paramecium has contractile vacuoles that collect excess water and pump it out of the cell. There are two organelles that are not part of the endomembrane system: chloroplasts and mitochondria. Both contain some DNA and ribosomes and make some of their own proteins. Both are fuel processors that can convert energy to forms that living cells can use. Most of the living world runs on the energy provided by photosynthesis. Chloroplasts are the photosynthesizing organelles of plants and protists. It's a solar power system that is much more successful than anything yet developed or produced by humans. The chloroplast contains the grana or granum. Granum is singular, grana is plural, and it's a stack of thylakoids. This is really a solar power pack, and it is where chlorophyll traps the solar energy. It is made of discs called thylakoids, so each individual disc is a thylakoid, and the chlorophyll is contained in the membrane of the thylakoid. It has an outer and inner membrane, and the space between the stacks of thylakoids, or the grana, is filled with a thick fluid called stroma. The stroma is where some of the chemical reactions for photosynthesis occurs. It contains DNA, ribosomes, and enzymes. The mitochondria is the organelle that converts energy from one chemical form to another. It carries out cellular respiration, which is when sugar is converted to ATP. It also has an outer and inner membrane. The inner membrane is highly folded that forms something called cristae or crista, which is singular, and those accordion-like folds increase the surface area for more cellular respiration. The mitochondrial matrix is the stuff in between the cristae, and it is where most of the chemical reactions of cellular respiration occur. It is important to remember that plants and algae have chloroplasts because they do photosynthesis, but they also have mitochondria for converting the sugar into ATP. Non-photosynthetic organisms need to consume food as their source of sugar for cellular respiration. The cytoskeleton is a supportive network of fine fibers in eukaryote cells. It is in the cytoplasm. It is for support and cell movement in general, but it may also mechanically transmit signals from cell surface to the interior of the cell. There are three main kinds of fibers that make up the cytoskeleton. Microfilaments help cells change shape by adding subunits at one end while losing subunits at another. They are solid helical rods made of actin, a globular protein, that forms a twisted double chain of actin molecules. Movement of amoeba and white blood cells involve the process of adding subunits from one end to the other. Microfilaments are also involved in cell contraction, for example, muscles. Intermediate filaments are a varied group made of fibrous proteins rather than globular ones. They have a rope-like structure. They serve mainly as reinforcing rods to anchor organelles. For example, the nucleus is held in place by a cage of intermediate filaments. Microtubules are straight, hollow tubes made of tubulins, which are globular proteins. They come in pairs, and the microtubule elongates or shortens by adding or subtracting subunits of those tubulin pairs. They have a lot of different functions, including providing rigidity and shape. They may assemble and disassemble where needed in the cell. They provide anchorage for organelles. They can act as tracks for organelles to move along. For example, a lysosome might move along to find a food vacuole. They also guide the movement of chromosomes during cell division, and they are the main component of cilia and flagella. Cilia and flagella are locomotor appendages that protrude from certain cells. They have a common structure and mechanism of movement. Cilia are short and numerous, and they usually cover the cell or the exposed surface of the cell. They are for moving cells or to move things over the surface of the cells. For example, a paramecium is covered with cilia that help to move food to its mouth-like pore or to help it move through the water. Flagella is longer and less numerous. There's usually only one or a few. It is for movement by whipping or propelling, and sperm is an example of a cell with a flagellum. Both are composed of a core of microtubules wrapped in a plasma membrane. There are 9 microtubule doublets and 1 pair of microtubules in the middle. At the base of the cilia or flagella, the doublets are anchored to a basal body. A basal body is 9 microtubule triplets. Basal bodies are identical in structure to centrioles, which are used in cell division. The basal body acts as a foundation for growth of cilium or flagellum. The doublet microtubules have little dining arms, which are knobs of protein that grab adjacent doublets and walk along them, causing the microtubules to bend, which creates the whipping of the flagella or cilia. In plants, cell walls protect the cells. Animal cells do not have cell walls. They provide skeletal support. They are 10 to 100 times thicker than the plasma or cell membrane, and they are made of fibers of cellulose, which, as you know, is a polysaccharide embedded in a matrix of other polysaccharides and proteins, similar to the structure of fiberglass. Between the walls of adjacent cells is a sticky layer of polysaccharides that glue the cells together. There are cell junctions that connect the cells to each other. The plasmodesmata are channels between the adjacent plant cells that form a circulatory and communication system connecting the cells and plant tissues. Water and small molecules can pass from cell to cell. In animal cells, the extracellular matrix is secreted by the cells, and the cells are embedded in it. It holds the cells of the tissues together, and it also plays a role in regulating cell behavior. There are 3 types of cell junctions connecting animal cells. Tight junctions bind cells together, forming a leak-proof sheet. Tight junctions line the digestive tract, for example, preventing fluids from leaking out. Anchoring junctions attach cells together and to the extracellular matrix. They basically rivet cells together with cytoskeletal fibers. Communicating junctions are channels that allow water and small molecules to pass from cell to cell. They are common in embryos where chemical communication between cells is essential for development.